Okay, so that's a quick overview of the drug development process. And again, our speakers will tell you more about it later on. I want to now talk specifically about the randomized clinical trial design and why that's so important, and how that and the analytics are actually going to make a difference in terms of the risks for investing. Part of what I'm going to describe is discussed in the article that was assigned for today, a primer on clinical trial design by the authors listed here. This is written for a very specific audience in urology. We're not urologists here, but it's actually a very clear statement about how clinical trials are put together. So I thought it'd be very useful to use that as a basis for it. So the basic idea behind a randomized clinical trial, I think all of you know, it's really meant to be a kind of a controlled experiment where you try out a particular therapy on a group of patients, but you also have a control group to see whether or not the placebo effect is really driving your results or whether it's really the drug or therapy. So it's also meant to be a prospective study. That word you will hear often in reading the literature because it's meant to contrast with retrospective study. A retrospective study is what we call data analysis in finance. It's looking at a historical record and trying to make inferences from it. It's very dangerous to use historical data because we can read all sorts of interesting patterns into the data, but in fact what matters is what happens going forward. So a prospective study is one where we say today, I want to know what the therapeutic value is for this particular drug. And so today, starting today, I'm going to do an experiment over the course of the next 2 years to see what will happen. So the idea behind the design is you're not using ex post justification. You're deciding today to study something that will happen in the future, and you don't know what's going to happen. You're not rigging the study. At least you shouldn't be rigging the study. And so that's the intention. It's said to be the gold standard of analysis in biomedicine precisely because it does control for this kind of backtest bias or this overfitting type of an issue. And we'll come back to it. I'm going to give you an example of that in a few minutes. The basic idea is to start off with a sample of patients that you divide in half. Half of them get the treatment, half of them get a placebo, or they get standard of care, depending on what kind of trial you're doing. And at the end of the trial period, you compare the results of those two groups. Hopefully, the group that gets the therapy is going to do much better in whatever metric you decide in advance, and the group that doesn't will not do as well. That's the objective of developing a good drug. And of course, if you can't tell the difference between those two groups, then you don't have a drug. Okay? That's the basic idea. Now, oh, yes, question?
If you're comparing to standard of care and you see no harm, like you typically see an approval?
Well, again, that's something I should ask our speakers here today, but let me give you a quick answer, which is it depends on the purpose of the trial. If the endpoint is, if it's a non-inferiority trial where you're not looking for improvements, but you're looking for less toxicity, or for example, the trial is meant to test out whether or not a dose of once a day is actually just as effective as 3 times a day, then no, you don't need to show improvements in order to get it approved. But you need to specify in advance that that's what you're looking to do. You can't, after the fact, say, gee, you know, that's sort of what we were looking for all along. That's right, that's what we wanted to do. Please approve it. It has to be something that was intended.